Okay, so for the third and last part of the Chapter 13 lecture, we're going to look at how viruses can cause cancer. There are a lot of things that can cause cancer, and infection by certain types of viruses is one of the things that can start a cancer. And so you probably remember that cell division, mitosis, the cell cycle, is under strict control. There are genes that are needed to activate cell division, and for most of the part, these are turned off in adults. And there are genes for stopping or inhibiting cell division, and those are generally turned on. And what we mean by turned off and turned on is they are turned off means not being expressed, no transcription and translation of those genes. And turned on means they are being expressed. So the proteins that are produced from transcription and translation of those genes has a certain function in cell division. So some proteins activate cell division, some stop cell division, and so those genes are controlled in normal cells. The term neoplasia, neo means new growth, new cell division. Typically, we use that term when there's an uncontrolled, somehow abnormal cell division, and it creates a whole bunch of cells, which is a tumor. So tumors start with just one cell, and that cell divides and divides and divides, and that forms a large mass of cells called a tumor. Then you have some language, the word benign. In general sense, the word benign just means harmless, but some tumors are benign but not cancerous. Some tumors are benign but will become cancerous. So this is where you need a specialist, an oncologist, to look at the different tumors that someone might have to determine if they think they're cancerous or not. But since we're focusing on cancer on this segment, let's talk about benign tumors that eventually will become cancerous. And what typically happens is as the cells continue to divide, they start to become more abnormal, and they can eventually spread out of their original site, so their initial site, and they can move, those cells might move to other tissues and start new tumors there. So if you have a tumor of lung cells in the lungs, then that tumor has not spread. But if you have a tumor made up of lung cells, but you find it in the liver, that would mean that the lung tumor had spread and started a new tumor in the liver. So a pathologist can tell what type of cell it is, and then by piecing together the location where the tumor was found, they can tell whether the tumor has metastasized. So metastasized means spread. So the term malignant and metastasis go together. That refers to a spread. Now, like I said, not all cancers are caused by virus, but virus cause a certain amount of cancer, and it's a fairly significant amount. And there's different ways that viruses can cause cancer. Some viruses have genes that are mutated, because you may remember viruses mutate quite a bit, and so they may carry some genes that have mutated, and when they inject or insert those cancer genes into the host cell, it can actually start that cell to become a tumor. Some viruses can insert next to the host cell's genes. So you remember how viruses can become dormant. They can their chromosome can be integrated into the host cell's chromosome, and sometimes when the virus is inserted into the host cell's chromosome, it causes overexpression or underexpression of genes that are really part of the host cell's chromosome. So the virus affects gene expression of the host cell. And then some viruses interfere with some of those genes that typically repress tumors. So if a virus interferes with tumor repression, what that means is it activates tumor formation. But there's different ways that viruses can cause cancer. And some of them, like human papillomavirus, has been very well advertised, has been very well promoted in medicine right now because we have a vaccine for it. And so if you get the vaccine, it doesn't prevent you from getting any kind of cancer, but it would prevent you from getting the kind of cancer that particular virus, HPV, causes. All right, so culturing viruses. The way we grow phage have to be grown in their own host cells, which are bacteria. And when you grow a phage, you have to then infect a culture of live bacteria. And you can spread that mixture on a plate. And you see a picture here of what it looks like on an agar plate. The bacteria create a solid smear of growth called a lawn, L-A-W-N. And then you'll have these little spots of clearing, little clear spots on the lawn, and that represents where the cells have been infected by the phage and then lysed, L-Y-S-E-D. So the clear spots are called plaques, and they're conceptually it's like the idea of a colony, but a plaque is a spot where you have phage growth, and they're so small that's why it looks clear, but there really are phage there. You may remember how small the phage are in comparison to the size of the bacteria. So the phage are there, they're just so small that it looks like there's nothing there. It looks like a little clear spot. And that's how you can grow individual phage on a plate. Now you can grow viruses. Animal viruses are typically grown in chicken eggs that have embryos, so fertilized chicken eggs. And it's pretty cheap to do. It's pretty cheap to get fertilized chicken eggs. And these are very large, fairly large cells. The egg itself is a sterile environment, and so they inject the virus into the center of the embryo and they can grow the virus. And that's why some vaccines have inactivated viruses that have been grown initially in chicken eggs. And so they sometimes ask you if you're allergic to, egg albumin, before they give you the vaccine, because it would have some of that material still in the vaccine. Here's a picture of a fertilized chicken egg. Here's the embryo here. They can either inject it directly into the embryo itself, they can direct it into one of these other membranes, they can inject it into the yolk. So it just depends what their procedure is in particular for that virus growth. And they figure out which location gives them the best growth for certain types of viruses. The last two topics in this chapter are really, really weird things that are sort of like viruses, but they don't have all the components of a typical virus. So remember, a typical virus has an RNA or DNA chromosome, and then it has a capsid made of proteins that surrounds that chromosome, and then it may or may not have an envelope. Then there are these things they have found called viroids, and these are just RNAs. This is going to sound crazy. It's just a specific small piece of RNA that has certain genes on it, that cause disease in plants. Now, do these exist in animals? We don't know. But the ones we have studied in science infect plants. They don't have a capsid, they don't have an envelope. The RNA doesn't seem to encode any proteins, but it has an effect on the plant, and it results in a disease. So in this picture on the left, some kind of potato that is normal, not infected by this viroid. On the right, these potatoes come from plants that have been infected. Now, if you ate the potatoes, it probably wouldn't do anything to you, because things that infect or affect plants, either way, would be unlikely to affect animal cells. So, but these have been found in plants, and I wouldn't be totally shocked if we eventually found these in animals, because a lot of things that we find in other organisms tend to also exist in animals. It's just we haven't found them yet. This is the small potato tuber viroid. So just to give you a sense of size, the chromosome of a phage, just by comparison, the phage is this really long strand. It's the phage chromosome. Actually, yeah, the phage. It's probably the DNA. Phage has a DNA chromosome. But then here you can see this potato spindle tuber viroid is just this small little segment. Oops, sorry. It's just this small little segment right here, or right here. So I don't even know how they ever found these. I mean, for Pete's sake. It looks like it could just be lint on the slide, but they know it's made of RNA. But man, it's small. Something else that's really odd, and this thing does affect animals, are called prions. Prions, the full name of them is Proteinaceous infectious agents. And they're strictly made of protein. It's a very specific kind of protein. All mammals have this kind of protein, but there is a version of the protein that has a different shape. So you can look on this image here on the left. On A, it says cellular PRP. That's the cellular prion protein. That would be a normal protein that everybody would have. On the right, on part B, is the prion protein, and what you can see is it has a different shape. That's what you're supposed to see there. It's the different shape of that same protein. And what happens is, if the prion protein version gets into your cells, that protein makes your normal prion protein change shape. So it makes your cellular protein refold into the prion shape. So this is very weird, because it's just strictly protein. There's no RNA, there's no DNA. It's just a protein that causes this effect. Now, what diseases are caused by prions? Spongiform encephalopathies. So spongiform refers to a spongy appearance under the microscope. Encephalopathy would be some abnormality of the brain. Encephala means the brain, and apathy means some kind of pathology. The word path, P-A-T-H, means some kind of abnormality. So encephalopathy, the encephalopathies are the brain abnormalities. And so when you take all of these terms together, what it means is this prion, a particular one of these, causes the brain to turn into a sponge full of holes. So we call this mad cow disease in cows. In humans, it's called Creutzfeldt-Jakob. That's how you would pronounce that in the German, Creutzfeldt-Jakob. Or in English, Creutzfeldt-Jakob. And it makes these holes in your brain. So you get this prion protein, and what it does to the tissue is, you can see down here on the left is normal brain tissue. On the right, you can see the spongiform version, which is full of holes. And I mean, basically, it makes you go insane. So mad cow disease comes from, in Britain, the word mad means insane. In America, it means angry, but in Britain, it means insane. So it's an insanity. It causes a mental, complete brain breakdown. And the scary thing about this is you can get this if you eat or have contact with typically a cow that has this. I guess if you eat a person who has this, you would also catch it. But that would be unusual. But if you have contact with, mucous membranes of somebody who has it, it could transfer. But typically it's from eating meat that had this, because when they slaughter the meat, some of the tissue, I mean, it's possible that some of the tissues get contaminated and you can end up with meat that has some of this in it. And there's no treatment for this or for any prion disease at this point. So the weird thing about prions is it's really hard to inactivate them. A lot of the things we talked about in terms of sterilization wouldn't work. You can use incineration, so serious high heat, and then autoclaving and concentrated sodium hydroxide seems to work. And they have found some enzymes that seem to work, but it's really hard. So you have to be real careful if you're working with a patient who has this in terms of the tools that have touched that patient's tissues would be contaminated potentially with that prion, and then you would have to really be careful to sterilize those tools. All right, so that's the end of chapter 13, three parts. You should answer 3 quiz groups of questions. I hope you have taken careful notes. And I may let you use those on the exam. So keep that in mind. I will let you use them on the exam. How about that? All right. So this is what we're doing because of the special circumstances that we're in right now. So go ahead and do your little quiz group and make sure you keep those notes.